All right, so we're back. Hmm. All right, so here's the structure. On the left is going to be the slides. We're going to go through them. On the right, we're going to have my notes. And so this important. It's important to have my notes on the side just because he's always talking about how you only need to know what he talks about. So notes on the right are going to help us keep us on track and not use up valuable real estate in our minds for information we don't need to know. First, vocab. A virus is a minuscule acellular infectious agent having one piece or several pieces of nucleic acid, either DNA or RNA. It has to be either RNA or DNA. It invades prokaryotes, eukaryotes, and causes disease. Okay, so here's some characteristics. Cannot carry out any metabolic pathway independently. So when it's an extracellular state, neither grow or respond to the environment, cannot reproduce independently, recruit the cell's metabolic pathways to increase their numbers, and here's what I just mentioned, have an extracellular and intracellular state. I'm going to have a slower start here because I'm eating a burrito, but we will ramp up here once I finish. I'll check out the group chat here, see what's popping. All right, so let's see. Once viruses have invaded a cell, they take control of a cell's metabolism to create molecules of viral nucleic acid bioproteins, which they assemble into new viruses. So we're going to get into this later on basically what virus replication is, but once it gets into the virus, all it's doing is replicating. Biological viruses are like computer viruses in that they are basically a code that is inactive and harmless until it infects its host. And it's devastating. Viruses have extracellular and intracellular state. So here's a photo that kind of showed the size. This red blood cell looks huge in comparison to a bacteria, and this bacteria looks huge compared to a virus. So here we have a kind of a bigger virus here, smallpox, and a really small one would be bacteriophage, which, as we can see, is as big as a bacterial ribosome, which makes sense because it's only containing um, double-stranded, at most containing double-stranded genetic material. Right. So in the extracellular state, it's called a virion, and is a completely ineffective structure. So when it's just hanging out outside and it's just floating around, mm, that's what it's called. The capsin and that section around it is a protein coat, which surrounds the nucleic acid. The nucleic acid and capsid are also called nucleic acid and capsid are also called nucleocapsid, uh, which in many cases can crystallize. So if we see the word nucleocapsid, we just know that it's this structure in its entirety. Some viruses have phospholipid envelope. So we're going to talk a lot more about this, but it's just some good introduction. Outermost layer provides protection and recognition sites. So in these, in the envelope especially, and around the capsid, the outermost layer provides protection, basically to protect the nucleic acids inside the genetic information. And it also has receptor sites so that things stick to it and that it lets them in. In the intracellular state, the capsid is removed so that we have the nucleic acid, and the virus exists as a nucleic acid, it still referred to as a virus. So once it's inside, it's a virus. But people call them viruses when they're not in there either. So just another vocab. Genetic material of viruses show more variety in their genome than all double-stranded cells. Their genome than cells. Genetic material of viruses show more variety in their genome than do cells that are double-stranded DNA. Primary way scientists categorize and classify viruses is type of genetic material. So this is where the kind of the meat and potatoes of this lecture is understanding the differences. So viral nucleic acid may be DNA or RNA, and can be double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. May be linear and segmented, or single and circular. So it might look like a bunch of little tiny ones. Like think of the chromosome of a human, or it might be that of like a prokaryote. Remember the DNA is circular. The chromosome is circular. And so have much smaller genomes themselves because it's only going to code for replication of itself and not for all cell functions. All right, so let's just take a look at what my notes say. Viruses lack any kind of cytoplasmic membrane, cytosol organelles. They are not capable of metabolic activity. When they invade a cell, they take control of its cell's metabolisms, and they use that cell's metabolism to produce more viral molecules and nucleic acids and viral proteins so that it can assemble more of itself. And so here's like kind of the central dogma. They go in, they destroy the DNA, and then they become the DNA and encode to produce more viruses. And they don't do anything else. They don't produce anything. They just make more viruses. So the extracellular state is often referred to as when it's in an extracellular state, it's often referred to as an inert particle, DNA or RNA, surrounded by capsule. Oh, so when it's surrounded by a capsule, it's protected from the elements, and the glycoproteins make up the capsin, nucleic capsin, in other words. It looks like we're a little behind on my notes here. Some say that some have a phospholipid envelope, and they steal it from our cells and gives them immunity. Because it gives them immunity because once it has our cell's phospholipid bilayer, it's going to have the same receptors and chemical composition as our phospholipid bilayers. So it's just going to look like any other cell. And the way that it steals it is that, a virus is going to go through a cell wall and it's going to puff out, but it's going to take that and wall that it went through. So it's kind of like, if you think about, it's like if you had like a wall of slime and you poked anything through it. Once you poke your finger through the slime's going to encase your finger. And so that's the way it's stealing your phospholipid bilayer. Intracellular state, capsin is removed. Only thing that enters is the nucleic acid. So yeah, so the capsin isn't just kind of removed. The capsin stays on the outside where it then like falls off or stays. But all it does is it's just going to inject the nucleic acid. That's all that goes inside of the virus. Um, so now we're caught up. So here we have an underline. Most viruses infect only particular host cells, and so this is so generally, viruses are very specific on what type of cell they're going to infect, and very very specific on the cell. And just an example of this is that HIV only is able to infect us because they have the correct receptors for T helper cells in our immune system. So, that's a very specific type of T cell, just to show like, how specific it gets. And it won't bind to anything else. Based on receptors, West Nile viruses are all right. Due to precise affinity of viral surface proteins and glycoproteins for complementary proteins and glycoproteins on the host cell. And that's just because the receptors have to fit like a lock and key. Maybe so specific they only infect particular kind of particular host. Okay, so here we have HIV only affects T helper cells. Generalists. So generalists are like viruses that can infect many kinds of cells and different types of hosts. So maybe it's just an important word, and that's an easy word to remember. An example of this would be the West Nile virus that can infect different types of species and cells. Okay. And so basically all types of organisms are susceptible to some sort of virus. All right, so here we have examples: plants, bacteria having viruses, bacteriophage, and human HIV, just to show differences. All right. It looks like I have some extra notes here. So capsins provide protection for viral nucleic acid, means that so the capsids are doing the attaching here to to a host cell. So that's kind of the means by which it does that. The capsin is comprised of proteins, protein subunits called capsomeres. So capsomeres may be a single, multiple types of proteins. So it's the capsin's protein structure that's allowing it to attach. And so viruses are normally classified into their shape. And so I'm going to grab some water and I'll be right back. All right, I'm back. All right, so viral shapes, viruses can be classified by viron shape. So what it looks like when it's in the extracellular state. So three basic types of viral shapes. We have helical, polyhedral, and complex. So complex looks like the lunar lander. That's like the photo that we've been seeing where it looks like a little capsule and it has like feet. So let's take a look at that. Oh, looks like I got my some of my notes here mixed up. So capsins, we'll just go over this again. Provide protection for viral nucleic acid, means by which many viruses attach to their host, composed of proteinaceous subunits called capsomeres. So the capsomeres are kind of like are proteins, are symbols of proteins, and basically capsomeres are what allow the for varied structure. The capsomeres are just varied assemblies of proteins, and capsomeres make up the capsin, and so that's how it creates uh, a sort of lock and key to adhere to host cells. The shapes of virons. All right, so here helical. That one's easy to remember just because helix, circular, and coil shaped. Helical, and then the isohedral looks kind of weird. I think the polyhedral. This one is polyhedral here. This one looks it looks like, I don't know if you ever watched Stranger Things, but or are you familiar with Dungeons and Dragons, but they use polyhedral dice, which are 20-sided dice for the game. Complex, capsid. So complex is the lunar lander, like I said. All right, so here we go. Enveloped helical virus. So this is the helical virus isn't just in a straight line. Here we can see how it's coiled up inside that envelope. Helical virus, and here we see still helical, but oh, that's why it looks like that whenever I see like photos of viruses and they all look like strings. It's because it's a helical virus that's just long. All right, morphology of complex, so that's how they look like. All right, so here we are. A viral envelope. That looks like we're at the end of my notes, but I have the next set of notes lined up. Characteristics of viruses, the viral envelope. So this envelope that we've been talking about, I've been already mentioning kind of what it is acquired from the host cell during viral replication or release. Um, envelope is a portion of the membrane system of the host, so provides some protection from the host immune system. So uh, it's literally just a portion of the cell's membrane, and it's just taking it when it's leaving. And if it's the same membrane, has the same receptor, same everything, it's going to be recognized as a self cell. Composed of phospholipid bilayer and proteins, because our membrane is made of that. Some proteins are virally coded glycoproteins. Enveloped proteins and glycoproteins often play a role in host recognition. Enveloped viruses are more fragile than naked viruses, and that is because uh, that kind of sounds counterintuitive, but the naked viruses, having to compensate for not having an envelope, are just generally stronger. While envelope viruses, all their nucleic acid is really weak. So once you dissolve their membrane, um, they just the nucleic acids are so fragile they just fall apart. But the naked viruses are a little more are just a little tougher. So it's a little more complicated for them. So here we have most susceptible to alcohol, detergents, and desiccation. Desiccation, I think, is running out. Yeah, desiccation means the process of becoming thoroughly dried or the state of being extremely dry. So, naked viruses are more stable outside of the host than enveloped viruses, which is, counterintuitive. Uh, when they name viruses, okay. Okay, so we have a helical capsid and the envelope in the glycoproteins. And so that's, the glycoproteins are just going to be the receptors. Classification of viruses, International Committee on Taxonomy of Viruses. So here's how we classify viruses. So viruses, taxonomy being like, categorizing. Virus classification is based on type of nucleic acid, presence of an envelope, shape, and size. So type of nucleic acid is, again, RNA or DNA, single-stranded or double-stranded. Viral genera have only been recognized, viral genera have only been organized into families. Only 9 viral orders have been described. Kingdoms, division, and classes have not been described. And this is because not a lot is known about them, so relationship among viruses is not well understood. Some researchers consider viruses to be the fourth domain of life. But this is the day we had the huge debate about whether viruses were alive or not. So, so here are my notes. We got typically they'll describe it because the type of nucleic acid or about the family. So Picornaviridae contains small RNA viruses. So Pico being small. Disease that they cause herpes viridae causes herpes. So that's just some other examples um, of why it's called that. So order, family, genus, viral replication. So family names typically derive from special characteristics of the virus within a family. So here we have I just kind of went over this, so we'll skip that. So here we have some families. Don't worry about it. Order, family, genus. HIV has not been assigned one yet just because there's not enough information. Viral replication. All right. Dependent on host organelles and enzymes to produce new virons. So it's just in order for a virus to replicate, it's depending on the host organs. So it can be lytic or lysogenic replication. It's a good thing we're going over this because I don't remember this at all. Lytic replication. Lytic replication is viral replication usually results in death and lysis of the cell. So here are the 5 stages of lytic replication. And another way you can remember the difference between what lytic and lysogenic does is that lytic is 3 words, and so is lysis. Not 3 words. It's 5 letters, and so is lysis. And so the first 2 letters are also the same. So it just looks alike mostly. So that's how I'm going to try to remember it. 5 stages of lytic replication. Of course, first one's going to be attachment of virus to host, entry of viron, so attachment, then entry, then synthesis of new nucleic acid and viral proteins, assembly, and then release. So there's some notes about that, but I think it's pretty simple. Here I have some notes here. Attachment, so entry of the virion into the host cell uses lysosomes, lysozymes, and so that burrow that those enzymes break away at the cell wall, whatever they're trying to get into. Right, let's continue. Attachment. Viron intact outside of the cell, inner attachment proteins attachment proteins touch the receptors has to be perfect, so may not bind to cell walls. Burst time. Okay, so a good term to remember is burst time, which is how long from entry, from attachment and entry for it to be released. So how long it takes to burst. Burst time kind of reminds me of Alien, the movie Alien. How long it takes for the alien to burst out. Right. So lytic replication. So here we have, it lysing here, like it exploding. So lytic bacteriophage replication. So attachment. Bacterial phages are non-modal. Oh, they can't move. Contact with bacteria occurs purely by random collision. So it's very random. These viruses can't move by themselves because they don't metabolize at all or adapt to their environment. Contact with bacteria occurs with purely random collision. Tail fibers responsible for attachment of T4 phage to host. So attachment proteins on tail fibers fit precisely into receptors for protein E. coli. So attachment proteins can be anywhere, even on the flagella or on pili. Uh, yeah, receptors can. So like for a host cell, it might not even bind directly into a cell wall. It might actually land on a flagella or pili, which is kind of interesting because you wouldn't think that would work, but I guess it can figure it out. Tail fibers responsible for attachment. Mm, all right. Tail fibers, toes, entry. All right. So it looks like we're just gonna go over this really quick. All right. So entry, it's gonna attach, then it's gonna enter. So lysozymes. Synthesis, after losing its chromium, ceases synthesis of its own molecules and synthesizes only viruses. So a part of this I haven't really spoken yet about is how viruses destroy the bacterial DNA at first. They destroy it so that it can replace all that information with itself. So synthesis, everything stops except viral replication. For double-stranded DNA, phage like T4, synthesis is straightforward and similar to cellular transcription. So yeah. Assembly is not well understood. However, it appears that as the capsomeres are assembled and accumulated in the cell, they spontaneously bond to one another to form new capsid heads. So it seems as if they self-assemble. They just like make all these parts and they self-assemble inside of the cell, which sounds crazy, but phospholipid bilayers form by themselves as well. So sometimes it's just the lowest energy state that makes it do it by itself. Release. So newly formed phage are released as viral lysozyme completes work on bacterial cell wall and bacterium disintegrates. Release of phage causes holes known as plaques. So the plaques are the holes. What? Newly formed phage are released as viral lysosome completes work on bacterial cell wall and bacterium disintegrates. So it kind of makes holes all over the bacteria. It makes it look like it's eaten, like holes bitten out of it. So burst time, we know that. So I'm just reading over the slides here. All right, so now, okay, so here we have the number of infective virons in medium. So here there's a lot, and then it goes to 0, and it's because all of them are going into a host. And then it's burst time, so now they're gonna get released. So here we have 1. So there's 0, and then there's 1. So then that 1 is going into the host cell, so that's why there's 0. For some reason, I thought this was a number bigger than 1, but, we're rocking with 1. So 1 makes 100 to 200. Viral replication. All right, so now we're on to lysogenic replication. All right. So lysogenic replication of bacteriophages, modified replication of cycle. So this is a modified version, it seems. Viral DNA incorporated into host chromosomes, allowing for viral DNA to be replicated alongside with host DNA. So instead of lysis just destroying the bacteria first, destroying the bacterial DNA first, lysogenic is kind of more stealthy, and all it's doing is implementing its own viral DNA alongside it, and then the infected host cell grows and reproduces normally for generations before they lyse. So this is kind of a more covert mission. So temperate phages. Yeah, I'm gonna ask something really quick. All right. So viral DNA is incorporated and before they lyse, so they lyse later instead of it just. So the the infected host is able to replicate by normally, and those other cells that it replicates are also infected. So temperate phages, prophages, and active phages. Lysogenic conversion results when phages carry genes that alter phenotype. So lysogenic conversion would be, the converting of the host cell's DNA. All right. Introduction. Lysogenic occurs from changes in phenotype in bacteria. Let's read my notes. Lysogenic replication doesn't destroy the DNA. Virus goes into genome and stays and becomes one with the bacteria. I like how I wrote that. Makes it itself resistant to other phages. So that's pretty interesting. So it makes itself resistant to other phages so that it is, you know, it called dibs first. Codes for something to make a harmless bacteria a pathogen. So here's just some notes I have on what makes them kind of dangerous. Is that often viruses can change the DNA and make what used to be a harmless bacteria into a pathogen, and that's why kind of some viruses, because viruses aren't inherently doing anything wrong. So why do you get sick? You know, it's 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 these steps. They're taking up energy. They're just diminishing the cells that they're infecting. But what else are they doing? And it's to make people so sick, and it's that it's coding. It can code to make harmless bacteria pathogenic. Here's what we have: bacteriophage, whatever that shit means. Lysogenic replication. So viral DNA enters the host cell. However, host cell does not destroy DNA. Phage DNA does not immediately assume control of cell known as prophage. So a prophage is an inactive phage when it's like just in there in the weeds. So prophage remains inactive, referred to as a prophage, by coding proteins that suppresses prophage genes. Oh, okay. So it's in this case, it's laying dormant. It's while it's in its prophage stage. Pro meaning before, so that's a way to remember what prophage means, or temperate phages. Um, so it's in that time where it's inactive, it's coding for proteins that suppress the viral replication, the prophage genes. Side effects of repressor protein is that it renders the bacterium. A side effect of the repressor protein that it's giving out is that it renders the bacterium resistant to infection by other phages at the same time. So that's actually good. Prophage for them. Prophage inserts DNA into bacterial chromosome. One piece DNA and becomes physical part of bacterial chromosome. All daughters of the lysogenic cell are thus infected with the phage. So, all right, cool. Phage maintain the bacterial chromosome. All right, so now we're on to lysogenic conversion. Lysogenic conversion occurs when a prophage changes the phenotype. Oh, when the prophage changes the phenotype of bacteria. So that's actually more relevant than what I said earlier. So, for example, from a harmless. Never mind. Oh, wait, I just remembered Savannah asked me to do something. Lysogenic conversion occurs when the phenotype of prophage changes the phenotype of bacteria. For example, from a harmless bacteria into a pathogen. So a bacteriophage actually might go into. I think I understood this wrong actually. So forget what I said earlier. So a bacteriophage would go into a bacteria that is like resident flora, just normal flora in your body, and then make it pathogenic. So bacteriophage genes are responsible for toxins and other diseases evoking proteins found in bacterial agents such as cholera. So maybe there's some form of some bacteria that lives inside you. It gets this virus and makes you sick. Oh, induction. So induction is the phage DNA moves away from the cell DNA. From my notes here. Point at which lysogenic phage is exercised from the bacterial chromosome and re-enters lytic phase. So now that it's been able to go through some generations, now it's gonna induction is the point where it's like, all right, let's just get out of here now and lyse in the lytic phase. So inductive agents are typically the same physical and chemical agents that alter DNA. After inductions, lytic steps of synthesis, assembly, and release resume the point. So inductive agents, what causes this induction, can be UV, X-rays, or carcinogenic chemicals. That is so interesting. I like how I'm reading through these and I'm like finding out new things that he didn't really go into in depth on. Like I'm learning the slides again. Uh, Lysogenic replication of in bacteriophage. So here we have lysogenic attachment, and it's gonna replicate itself, replicate itself, replicate itself. So this would be the lytic cycle, but the lysogenic cycle would be when it's it's laying dormant in there, letting itself divide, and then it's going to infect more. I wonder which one is more effective. So break time. All right. So replication of animal viruses. Now we're on to animal viruses. Same 5 basic steps for their replication pathway as bacteriophages. So that whole time we were dealing with bacteriophages. So only affecting bacteria. So sorry if that confused you, but now we're on to animal viruses. So it has the same 5 basic steps: attach, entry, synthesis, assembly, and release. And differences result from presence of envelope around some viruses, because now envelopes are going to really matter in this sense, because our cells don't have cell walls. Eukaryotic nature of animal cells and lack of cell wall. Yeah. So there's going to be some differences. So attachment, dependent on chemical attraction and exact fit between viral protein and the cell receptor. So the virus is going to have an attachment protein, and it's going to bind to a cell's receptors. And that's the key and lock and key feature. Animal viruses do not have tails or tail fibers. All right. Replication of animal cells. Entry and uncoding of animal viruses. Animal viruses enter host cells shortly after attachment. There are at least 3 different mechanisms in which animal viruses penetrate. All right. So first of all, it's direct penetration, which is where the capsid sinks in and creates a hole. And then the capsid opens up, and then the DNA is released. So that's like if you drop the rock on some dirt and it makes a little crater around itself. So that's how it's going to sink into that cell wall. But what I didn't mention here is that indirect penetration, there's gonna when the capsid opens up, it's also penetrating into the cell. So it's like going to directly prop it in. I think there's enzymes that aid in that. Let me look it up. Do enzymes aid in naked viruses? I don't know. But basically, it's just direct penetration. It's just going to sink into it and bury itself in there and inject itself. Membrane fusion is that since for enveloped virus, since it has the same cytoplasmic membrane as the cell that it's invading, sometimes it it would just fuse with the membrane, and it'd kind of be like endocytosis, where you got like a blob, and it's just going to join with the cell rest of the membrane. Endocytosis would have this is this would be an example where a virus has like certain proteins that encourage the cell to swallow it. So it's kind of tricking the cell into doing the work for it. All right. So here we have photos. So receptor. So receptors. It attaches and then it injects itself. Here we have the membrane. Or this is it being swallowed. Oh wait, never mind. This one. So this has the same membrane, and all it's going to do is see it's binding, and then it's just opening itself up. And for endocytosis, these receptors and proteins are just encouraging it, through signaling or whatever. I don't know how they do it, but it's basically telling it, "Hey, eat me. I'm something that you want." And so that's how it does it. So here we see that the capsin actually makes it inside. Which is weird because I thought in a previous side that I said that the capsin does not go inside, but whatever. So direct penetration. Used by some naked viruses, capsid attaches and sinks into cytoplasmic membrane, creating a hole or pore in the cell membrane. Viral genome alone enters through the pore, leaving the empty capsid on the surface. That's what I'm more familiar with. I don't know why we see it not that. Oh wait, never mind. We do see that. Let's see what it says for the other stuff. Okay, so for membrane fusion, host cytoplasmic membrane fused with viral envelope, capsin released into cytoplasm. Okay, so that does happen. Leave envelope glycoproteins as part of cell membrane. So the membrane is just being left there just because it's fusing. So it's pretty interesting. And so endocytosis is used by enveloped or some naked viruses. I don't know what the point of saying that is because there's either enveloped or not enveloped. So I don't know why. Most enveloped and some naked, because the ones that are enveloped are going to have that bilayer. So viruses attached to cell receptors stimulates the cell to absorb the virus. So it's just telling the cell to engulf it. All right. All right, so now we're at DNA synthesis. Synthesis of DNA viruses of animals. Each type of animal virus requires different strategy depending on its nucleic acid. DNA viruses often enter this nucleus. DNA viruses often replicate. DNA viruses often enter the nucleus. RNA viruses often replicate in the cytoplasm, just because that's RNA leaves the cell, so it would act on the cytoplasm where the rest of the RNA is. All right. So, based upon how mRNA is synthesized, what serves as a template for nucleic acid? All right. So, synthesis of DNA, we have double-stranded DNA viruses. So now we're going to go through the whole shebang. So this is similar to the replication of cellular DNA. So how is it? How is it going to grow itself? So hold on, I'm gonna take a 30-second brain break. All right. So double-stranded DNA viruses are similar to replication of cellular DNA. Viral genome replicated in the genus. Viral genome replicated in the nucleus. So it's just going to be replicated in the nucleus alongside the other DNA. Because now we have nucleus. Because now we're dealing with animal viruses. Viral mRNA synthesized from the viral DNA, and so that mRNA is going to go to the ribosomes and just tell it to make proteins for virus synthesis. So this is the most straightforward way. The DNA infiltrates the rest of the DNA, and then it just from that DNA polymerase is going to code viral mRNA. So viral capsomer proteins are made up in the cytoplasm and migrate into the nucleus where new virions spontaneously assemble. So this is like a papillomavirus or herpes. So this one's pretty simple to understand. Hepatitis B virus replication. Some exceptions. So there's some exceptions to this rule. Hepatitis B virus replicate from RNA intermediary instead of replicating DNA from the DNA template. So genome is transcribed in RNA. I don't think that's important, but I'll just go over it quickly. Hepatitis viruses replicate DNA from RNA intermediary instead of replicating DNA from a DNA template. So genome transcribed into RNA used to make multiple viral DNA. So in this case, RNA is used to make DNA, which is not what normally happens, and this is only possible because of an enzyme called reverse transcriptase, which the virus carries. So that's actually important. Never mind, I just didn't read it right. So just know that reverse transcriptase is used by some viruses in order to turn RNA into DNA and infiltrate that way. Pretty interesting. All right, so now we're on to the single-stranded DNA virus. Synthesis cells do not use single-stranded DNA. It's all double-stranded, so they have to make a workaround. And in this case, all it does is DNA strands folds back on itself to make a hairpin loop and acts as a double-stranded DNA, which is replicated by cellular polymerase, and so then more viral DNA uh, is being produced. Really replicated strand is released as single-stranded RNA. Single-stranded RNA packaged into viral capsid, cellular RNA polymerase. So basically, DNA folds back on itself in a hairpin loops and acts as DNA, which is then replicated. So yeah. And then, yep. All right, so synthesis of RNA viruses. So there are 4 types of RNA viruses. There is a positive single-stranded RNA, retroviruses, negative single-stranded RNA, and double-stranded RNA. Okay. So what does that positive mean? Synthesis of RNA differs from DNA virus replication. Since DNA is already like the head of the cell, it's kind of easier for them. So the RNA ones are a little more complicated. So a positive sense, viral RNA acts as mRNA, and that works well just because if it acts as mRNA, it's able to code for proteins, and make more viruses. So yeah, that's why it's a. So this is where that little plus comes in from the single-stranded RNA. So then there's also negative, which is viral DNA that cannot be translated to mRNA. So that's just the difference. Plus means that it can act as mRNA immediately, and negative means that it cannot, but it will, but not immediately. So negative negative sense single-stranded RNA have to have a couple other steps that we're going to go over. All right, so here we have. Oh, so here we actually have the example. So single-stranded RNA, the capsid single-stranded virus is delivered by a capsid. Um, it injects itself. So this is a form of practicing. It looks like it's going to be direct penetration as a form of entry for animal viruses. So, and it just drops off a single-stranded RNA. So it just uses RNA polymerase, and it makes a complementary SS RNA, negative one, and it makes it just acts as a translation. Yeah. So here's how. Sorry, uh, tripping over my words. Here we have two pathways. One of them, since it's a positive single-stranded RNA and it's able to act as mRNA, it here we have the translation of viral proteins. It's just making protein since it can act as mRNA. mRNA is used to make protein, and in order to keep on replicating itself, it it makes a negative strand of itself, which makes a positive strand of itself, and it just keeps on going. And so when it makes a positive strand of itself, with the nucleic acid on the left side and from the right side, all the proteins and basically like the maybe the capsin for it, the capsid, it assembles them. So here we are. It's a little more complicated for negative stranded RNA, and it's that it has to transcribe itself with RNA dependent RNA transcriptase. So dependent, whatever comes before dependent is whatever it's acting on. So RNA dependent means it's acting on the RNA transcriptase, where it's going to form a complementary positive sense RNA that acts as a template for as mRNA. And so once you have that positive sense, you're just going to make viral proteins, and then you're going to transcribe uh, to make the negative single strand RNA. And once you have your negative stranded RNA again, another one, and your proteins, you're able to assemble more. So yeah, you guys can pause and take a look at that to kind of get it. So in order for it to kind of oh, uh, for it to switch from negative to positive, all it has to do is transcribe. So here we kind of see that in action here. So but remember there was four, and one of them is double-stranded RNA. And so for double-stranded RNA, all it's doing, it's kind of a combination of the both processes, just because it simply unwinds, and then it has the same process for positive-stranded RNA and for negative. So it's gonna make itself. It's gonna make another one. Uh, so here we have positive stranded RNA acts as template to make viral proteins, and when for the negative strand, once it unwinds itself here from here to here, transcription of viral DNA polymerase can make complementary RNA strands, and they just make another one. So now you have your double-stranded RNA and your proteins, and it's going to make another virus. So that's kind of how it goes. So you have to consider it has to make a clone of itself and make the proteins for its capsid. And so, that's the general gist. So let's see what I have on my notes. Single-stranded DNA cells do not use. Never mind, I already read that. Positive single-stranded RNA. That's an RNA. Not be recognized, but so negative single-stranded RNA cannot be recognized by cellular machinery. So that's why it has to transcribe itself, and in order to be recognized, and it's going to transcribe itself again to make a negative, to make the opposite, the negative, and then it's going to send it into a capsid, and it's going to make a new virus. So it carries, so negative stranded RNA actually carries an enzyme. It carries the RNA dependent RNA transcriptase, uh, transcribe itself into a positive mirror. To find the ribosome. So that from being able to flip it from the negative to the positive, it's going to transcribe itself. But normally RNA doesn't do that. So that's why RNA dependent RNA transcriptase is needed for that. And so the virus brings that in. That's not part of the cell. So now we're on to double stranded RNA, which unwinds, and I didn't write anything else. Now we're on to retroviruses. So retroviruses are actually positive single strand RNA, but don't use their um, but they don't use their genome. They don't use their nucleic acids to act as regular messenger RNA. Retroviruses double what double strand DNA intermediary transcribed by viral transcriptase. I'll try to use the template to produce. Oh, okay. So double-stranded intermediary transcribed viral transcriptase. So basically, the positive single-stranded RNA in retroviruses uses RNA dependence, 'cause it's acting on the RNA, DNA polymerase, where it forms DNA out of the single-stranded RNA. So that's different. So that's a good way to think about it. Retrovirus, it's going backwards. So instead of going to DNA to RNA, it's going from RNA to DNA. So that's how it goes. Guys, I'm tired and I want to play video games. So double-stranded RNA uses a template to produce, and so from that, once it creates that DNA, that double-stranded DNA is used as a template to create more of the retrovirus. So you got to think about it each time, like this is what it's doing in general, but also how does it go back to making a clone of itself? How does it clone itself to make more? Because obviously its end goal is not just to go inside and transcribe itself and hang out. It wants to clone itself. So all right, so now we're on to negative sense ssRNA. So negative ssRNA viruses. Um, yeah, so this is just the same thing I already said. Each strand acts as a template for other strands. What? Double-stranded RNA viruses cause most diarrhea in infants. It's funny things to mention. Each strand acts as a template for other strand reminiscent. So yeah, double-stranded RNA, you can just kind of think of the same process. When is it going to transcribe? When it unwinds, because when it unwinds, it also has to wind up again. So. So here we can go over this just because this is kind of the meat and potatoes. So I'll go over this. Double-stranded DNA. How does it? How is mRNA synthesized? Because remember, we want to make protein, so mRNA is the goal. So for double-stranded DNA, bicellular RNA polymerase in nucleus or by viral RNA polymerase in cytoplasm. So All right, I read all that, so you guys can read that too. All right, so now animal viral release. So for yeah, I'll be back. Yeah, I would like to speak with you. I would like to speak with you regarding football things and the beautiful game. Is how much are you practicing them? What? Like injury, like for real injury, or like fake wussy? Oh, I like this type of season ending torn ACL injury. So it does sound like it's actually hurt. So well, you've also been animals and get to recovery. Eat lots of bananas so you can recover, and do not play on or do anything too crazy on that hurt leg. Because literally, yeah, but just don't cut it on too far because that's literally how chronic injuries happen. Is when people like once you hurt a leg, if you don't let it fully heal, even one time, it'll always be tweaked. So let it fully heal before you start getting too crazy. Otherwise, it'll just be. Otherwise, it'll have that injury for the rest of your life. Otherwise, it'll have a weird loosey cement. So that's my advice. I'm returning you to cement. So you're injured? Whatever. All right. So viral release. Enveloped viruses release through a process known as budding. I don't know how to remember that. Envelopes. Maybe it's you send your buddies envelopes. So envelopes like letters. It's kind of cute. As viruses are assembled, they are extruded through cells membranes. Um, oh, right. So never mind. Budding. Budding would be the process where like once viruses are like made and they're pushed through a cellular membrane, like that phospholipid bilayer, and they pick it up. That would be budding, which would make sense actually. Each enveloped viruses acquires a portion of the cellular membrane. It actually takes it into which viral proteins are inserted during the synthesis stage. So viral proteins are inserted into the cellular membrane so that it can continue to affect that specific host that originally infected with that same viral protein. So released. Naked animal viruses are released via extrusions from cells via exocytosis. So exocytosis is when it just makes a big clump, like yeah, with a cell vacuole, which is like a big clump of stuff, and just throws it out. And it has its own membrane. The vacuole is surrounded by a membrane. So, but this is different from an envelope one because obviously these guys aren't picking it up because they're naked. So, fusion of vacuole membrane with the outer membrane, and then so yeah, or lysis of cells. So, naked animal viruses either do exocytosis or lysis of the cell, and it seems that envelope viruses only do budding, where they just leave. All right, so persistent infections. Animal cells do not quickly lyse. Allows infected cells to remain alive for some time. Infection with animal viruses, which are shed slowly and steadily, are referred to as persistent infections. All right, so an enveloped so viral capsid. So here it's already been created, and the budding process is when it steals it, and then there you go, an enveloped virion. All right, all right. So let's look at my notes. Budding, if they are enveloped, they go through the nucleus, run a rough endoplasmic reticulum, or the cellular membrane, because these have phospholipid bilayers. The nucleus, the RAR. Hmm. So we have latency of animal viruses. Some animal viruses remain dormant in host cells in a process known as latency. Viruses that are called latent viruses are called latent viruses or proviruses. Hmm. Incorporate into cellular DNA. May be prolonged for years with no viral activity. Incorporation of provirus into host DNA is permanent. So single-stranded RNA viruses carries reverse transcriptase, enabling it to make a DNA copy, which they can be incorporated into a chromosome. So this would be an example of a retrovirus, as we just mentioned. So let's see what my notes have. Viruses, latency. Viruses that are called latent viruses if they take a long time, um, where they remain dormant. Hmm. All right. Under normal conditions, the cell division is under strict. Under normal conditions, the cell division is under strict genetic control. But viruses can cause cancer because they're ignoring. So there's parts of each cell, parts of each genome that protect against just rapid and uncontrolled cell division, which is what cancer is. But it could be possible that the virus interferes with that gene either by eliminating it or turning it off. And so that's why viruses can have a pretty direct role in cancer. So, under normal conditions, the cell division is under strict genetic control. Genes dictate that some cells can no longer divide at all. Cells that can divide and are prevented to from cells that can divide are prevented from unlimited division. Genes for cell division turned off or genes inhibiting turned off. So neoplasia is uncontrolled cell division in multicellular animals, so otherwise known as tumor. And so benign versus malignant. Uh, benign stay put and malignant spread out. Viruses go get there, viruses control the control cell. And so metastasis is when a tumor spreads. All right, which is kind of easier to remember because malignant means spreading. So malignant metastasis. So we're on to the proto-oncogene section. So proto-oncogenes promote cell growth and division, codes for proteins that help to regulate cell growth. Uncontrolled activation of oncogenes can lead to cancer. So that's what viruses can interfere with the proto-oncogenes and basically make it divide too much. Environmental factors that contribute to activation of oncogenes. Ultraviolet
light radiation carcinogens of viruses. And so the way that the ultraviolet light radiation, like, how is that actually making the proto-oncogene go more? Wouldn't it be inhibiting it? You got to think about also the inhibitor. There's inhibitors on our genes. Remember from the operon. If that inhibitor on the operon is damaged, then there is no inhibitor, and then obviously this proto-oncogene can run rampant. Yes. Yeah. So here's how, for example, it's showing. So virus inserts itself into the repressor gene. Exactly. Can't be repressed anymore. So it's just going to get crazy. Viruses may contribute to viruses may contribute to the development of 20 to 25% of human cancers. Wow. Some carry copies of oncogenes as part of their genomes. So that's not good. If they're carrying an oncogene, that means they're just going to insert it into a cell and make it, divide even more. Some interfere with tumor suppression, so that's another way. And specific viruses are known to cause human cancers, like cervical cancer, blah, blah, blah. Viruses. Oh, so it looks like we're done with that. All right, so now we're on to culturing viruses. Culturing viruses in the laboratory. Viruses cannot grow in standard microbiological media because they can't metabolize by themselves. So viruses must be cultured inside of host cells. So three types of media for cultivating viruses. Media consisting of mature organisms like bacteria, plants, or animals. So embryonated eggs is a way, or cell cultures where you just throw a virus onto a bunch of cells, bacteriophages. Culturing viruses in mature organisms, using bacteria as a virus culture medium. So phages grown in bacteria in liquid cultures or in agar. It's cool. So bacteria just needs to be alive. The virus just needs to be on it. Lysis of bacteria produces plaques. So once all that bacteria blows up because the virus is making it blow up with more viruses, it produces a plaque on the agar plates, and so the size and how many of them allows for estimation of phage numbers by plaque. Uh, viroids, bacteria used for culturing. Oh shit! I gotta plug in my thing. Sorry, I was messing with my lamp there. So, all right. Bacteria used to culture bacteriophages can be grown in liquid. Uh, Uninfected bacteria grow normally, and there's going to be some uninfected bacteria if it's a multiple types of bacteria in the agar plates, because remember, like viruses have to be very specific. So bacteria can liquid agar on agar viruses Mixed with warm nutrient can be poured into the line over to page by and infect bacteria, lice cells, infected. An uniform bacterial lawn interrupted by clear zone referred to as plaques. The clear zones are plaques. So here are our plaques. So you can estimate the number of phages. So we can also culture them in plants and animals. Numerous plants and animals have been used to culture viruses. Laboratory animals can be difficult and expensive to maintain and also maybe unethical. So they're not a very popular form of doing that. So culturing viruses and embryonic. So for embryonic chicken are inexpensive, among the largest of cells, free of containing microbes, contain a nourishing yolk. Fertilized chicken eggs. Some vaccines can be prepared in eggs. So interesting. Culturing tissues from cell tissues. So cells isolated from an organism or grown onto a surface of media broth. Cells cultures, sometimes inaccurately called uh, tissue cultures. Cell cultures are less expensive for maintaining animals and plants, and avoid moral issues that sometimes associated with experiments conducted on humans and animals. So if you want to, for example, for research culture HIV, you wouldn't want to give it to a human. Two types of cell, but it needs a host. So two types of cell culture. So there's diploid cell culture created from embryonic plant. Generally last. So he didn't cover this. Oh, he did. Oh shoot, I'm lying. Oh, we gotta go back over it. All right, so two types of cell cultures. There's diploid cell cultures from embryonic animal plant cells that have been isolated and grown under specific conditions. Generally last for 100 cell divisions before they die out. Continuous culture. Continuous culture, longer lasting, derived from tumor cells, neoplastic. What else? Are viruses alive? They're considered the complex pathogenic chemicals. Recall the four characteristics of life. Other consider them to be the least complex living entities, but which is weird because they have such sophisticated methods and evolve and replicate themselves. So it's like what are these things? How do we classify these things? So here we have some more characteristics of viroids: extremely small circular pieces of single-stranded. Oh, viroids, which is different from viron. So viroids. Viroids are extremely small circular pieces of single-stranded RNA that are infectious and pathogenic in plants. So viroids are for plants, similar to RNA viruses, but lack a capsid. So that's super interesting because that means they're literally just uh, nucleic acid particles just hanging out. Viroid RNA does not code for proteins. Hmm. Viroid RNA adheres completely to plant mRNA. So the viroid DNA adheres to messenger RNA, and that's how it infects it. So viroids are for plant. Viroids only affect plants. So they make double-stranded RNA. These are simple. Oh, So the viron RNA adheres to a complementary mRNA. So then they make a double-stranded RNA, but the plants don't use double-stranded RNA. So it dis- so plant enzymes destroy this and it makes it disease. All right, so now we're on to prions. Prions are prior to the discovery of prions, diseases that were incorrected as slow virus disease. Prions are associated with so-called mad cow disease. Some scientists suggest non-infectious prions may be also involved in Alzheimer's disease, Parkinson's disease, ALS, and even some types of cancers. So, what are prions? So, prions are proteins, but they're infectious particles. Um, and what they do is that they change non proteinaceous, not viruses because they lack nucleic acid. They are proteins. So, cellular PRP. So, we have cellular PRP in our brains, and they're involved in brain activity. Since so when they are PRP, they have a helices shape, but a prion for PRP causes the cellular PRP, but a prion would make these cellular PRPs, change shape into a B sheet instead of a helices. So prion PRP causes a cellular PRP to turn into more prion PRP, and it's just this protein that's going in that's not alive and just simply influencing the other proteins in our bodies to change shape. And by changing shape, it can uh, interfere with normal processes. Like for example, they found a link with ALS, Parkinson's disease, and Alzheimer's. So if it's in the brain, it's messing with something in the brain. So PRP acts as a bad influence in a crowd of teenagers, encouraging molecules of PRP to misbehaving and refolding and turning into prion PRP. As clumps of prion PRP propagate throughout the brain, neurons stop working properly and eventually die, leaving holes and spongy in appearance. So it just degrades it. Prions with amino acid occurs on. All right. So cooking and normal sterilization procedures do not deactivate prions. Prions destroyed by incineration or autoclaving concentrated. So they have to be incinerated. It's a new problem, a problem that we're addressing now. So it looks like there's some new action against it. So there you go, guys. That is all. So I will live stream the rest of this stuff that we went over um later. But so I gotta take a brain break. All right, bye guys.